Hello, everybody. My name is Susanne Weis, and in my talk, I would like to address both the chances and the pitfalls related to prediction of mental traits from brain imaging data. So I'm trying to make the point that, yes, we can be excited about the possibilities. We also need to know when to be critical. First of all, why do we even want to make predictions based on brain imaging data? Well, we are all different. Every one of us is different. We all have different mental attributes, different personalities, different performances, different cognitive aptitudes. And obviously, there's also differences in psychopathology. And while these are our personal traits, we cannot, as one person, we cannot easily infer information of the mental traits of another person, even though there's lots of situations where this might be necessary or useful, not only in the clinical case, but also, for example, in job interviews or in legal cases. One person needs to make judgment of another person's mental attributes, and these we can obviously not directly access. We can try to approximate them by using interviews or by using certain cognitive tests, but it's still only an approximation. And with the advent of better and better functional and structural brain imaging methods, there is clearly the hope that if we are using these data of the individual brain, which is clearly influenced by mental attributes, we might be able to make judgment about certain traits in a better and more accurate way than relying only on interviews or tests. Of course, the idea of examining individual differences is not new at all. And there's a large literature in many fields of cognition and neuroscience where we've seen group comparison, like this constructed one as an example. This would be a study that compared the performance of men and women on a certain cognitive task. And when we see these group comparisons, we see a highly significant effect here, women performing significantly better than women. But the question is, what does that, can this tell us anything about the individual, or can that be used as a biomarker? And clearly, when we look at a bit more honest presentation of the same data, we see that no, because all significance tests, group comparison, refer only to the observed means. So yes, there's clearly a difference in the group of females versus the group of males, but it does not allow for any conclusions on an individual. Though there's, for example, many men here that perform better than some of the women and the other way around. So mere group comparison of one variable surely does not allow for any inference about the individual. On the other hand, we have to be aware of significance is different from relevance. So we might have a case, again, a constructed example, where we compare baseball players with sumo wrestlers, and we compare them by their height, and we see there's actually no significant difference at all between the two groups. Similarly, for the weight, no significant difference between basketball players and sumo wrestlers. So it seems we cannot actually distinguish the individuals. But when we put both variables, so both dimensions together in a two-dimensional space, we clearly see a nice separation between the two groups that will allow for the assessment of individual differences. So multi-dimensional patterns can help us or can increase the predictive powers versus only looking at one single variable. But one thing that we need to be really aware of is in all these approaches are prone to overfitting. So if we would just look at these small sample and apply any conclusions to a different sample, we would probably not be able to predict very well, because our prediction might be too much based on this one. So we need out of sample validation before we can draw any real conclusions about unseen individuals. So, and clinical questions, so the application of the machine learning methods to clinical questions clearly is most interested in individual cases. And what we do usually in the application of machine learning methods, we use a training group, as big as possible training group. We extract some features from this training group, for example, functional imaging features. We clarify which target variable we want to predict or classify, and then build a model that uses the features to predict the target in this training data. And once we have a model that works well in the training data, it is absolutely essential that we also test this model in a completely independent test group to validate if the prediction of the target only works in the training group or as it should in the test group as well, to be generalizable. All these methods is a very short summary. All these methods are relatively complex. So luckily there's more and more toolboxes being developed. One of those is being developed in our institute, which make it a bit easier for the user to apply these machine learning methods in a correct way. Okay, let's come to a first example of successful predictions. A topic that has been addressed many times is estimating age from structural brain imaging data, so gray matter volumes. And in this particular study, the authors used 128 different workflows, so different feature space, 16 different feature spaces, meaning different ways to create features from these gray matter images, and 8 different machine learning algorithms. And with doing so, they could identify the best algorithms which allow for very accurate prediction of the predicted age as shown on the y-axis in relation to the chronological age from this gray matter data. And importantly, this was true not only within sites, so not only within the training group, but also across sites. So application of the model to a different sample from a different acquisition site. And furthermore, these predictions can be used again with a clinical application in showing that the difference in predicted age and actual chronological age is much larger in Alzheimer's dementia patients shown in red than in control subjects shown in green. And the difference is in between for early and late MCI. So basically this already is one good example to show that yes, machine learning can find generalizing MRI phenotype relationships. However, importantly, especially when dealing with brain imaging data, we have to consider the dimensionality of our data. So meaning that the algorithm does not actually see a brain, the algorithm sees, for example, if we're dealing with structural data, 400,000 voxels, so the algorithm sees 400,000 voxels, and it doesn't even see them like in this shaped in the shape of the brain. It actually just sees a vector of 400,000 numbers. And even if we have large cohort data, then we still face the problem that the number of features, so in this case, the 400,000 voxels, will be much larger than the number of subjects we have to train our model on. And this is a problem because with many more features than subjects, we can always train a model that predicts perfectly on this training sample, but will most likely not generalize well because it's only overfitted on this training sample. However, again, in the special case of brain imaging data, we have a relatively straightforward ways to reduce the pseudo-dimensionality because we know that the 400,000 voxels are not independent. The brain is organized in brain regions that are activated together or in brain networks that are activated together. And using this biologically informed data reductions can help us a lot in dealing with the dimensionality problem. One example for this was a study to predict sex based on functional brain imaging data. And what we did here, we used a brain parcellation, basically an atlas of the brain that has been defined based on different regions' connectivity patterns. And then we could use the connectivity between all these, in this case, about 400 parses to look at the whole brain connectivity. But we can reduce the dimensionality even more by looking at each parcel separately. So basically building separate models for each of our roughly 400 parcels to predict or to see how well sex of the participant can be predicted of this parsel's connectivity. And that has an additional advantage that we can also show which areas of the brain distinguish more between males and females by looking at the performance of the different models across the brain. The same approach is not only possible for classifications or binary classification, but it's also possible for the prediction of cognitive performance, in this case, executive function. So again, based on brain parcels, we can see which areas of the brain predict executive function performance particularly well. So altogether, individuals' prediction in new subject is possible from neuroimaging data, and this is applicable to a variety of different phenotypes. I have now shown you sex, age, cognitive performance, but it also works for example, clinical condition or psychopathology scores. Coming to another caveat, so as I mentioned, we need to be aware of generalizability, but that refers not only to the variability between different samples that we're looking at, it also pertains to more technical variability in the sense that if we train, for example, again, a sex classifier on data that was acquired on a particular brain imaging site on a particular scanner, we can achieve a model that predicts quite well, even across different samples from the same scanner. But as soon as we apply our models to different scanning sites, where the scan is the pure technical parameters are different, we will likely find that the models do not perform that well anymore. And that means for actual well-performing good models, we need training data from a variety of different sites. So we need to control for non-biological variability of no interest, by considering these batch effects and by training across different scanning sites. A different way of reducing dimensionality of our data is to use meta-analysis, networks defined based on meta-analysis, making use of the fact that we already have a huge neuroimaging literature, and we can compress all this knowledge into meta-analytical networks for different cognitive functions like language, working memory, emotion, anything that we want, basically based on appropriate meta-analysis. And then we can extract the nodes of this network and use these nodes to extract brain connectivity of each individual subject in data that was, for example, acquired in a resting state scan. So the subjects only lie in the scanner for 10 minutes doing nothing in particular, and then we can use the same data, the same resting state data, to look at features derived from a variety of different meta-analytical networks. And this can be shown to make sense because this is a study where random networks were constructed with an increasing number of nodes, and we see the more nodes the network has, the better is the identifiability of single subjects. But our meta-analytical networks perform better than any random network, with the exception of a motor network. And also we can use other brain states. We can induce brain states by, for example, instead of just putting participant in the resting state in the scanner, we can show them movies, and depending on the brain state that are created by these movies, these meta-analytical networks will react differently. So, but both with the movie data and the resting state data, we can, by using meta-analytical networks, extract a variety of different feature sets and do a variety of different predictions based on just one single data set. However, we need to be really aware of the characteristics of our samples, as this study shows, a study where hand grip strength was predicted from brain data, and it looks like the prediction works really well, R of 0.57, so it looks nice. But if we look at our sample in more detail, we see that actually, when we look at males and females shown in blue and red here separately, the prediction works much less well. And importantly, we see that the overall good prediction that we think we saw in the first picture is actually driven by sex rather than actually predicting hand strengths. So the quality of our models depends on targets and the feature space, and importantly on the characteristics of our sample. And the sample and the features and targets can also be vulnerable to certain biases, as showing here. Again, this is an example of a sex classification study based on structural imaging data, where a sample was created by matching males and females for age to control for age effect, and then extracting local gray matter volume features to try to predict whether a certain brain is male or female. And indeed, we can do this with a very high accuracy of 0.97. But when we look at the model output, we see a very biased model, because basically what we see is that both for the males and for the females, those individuals who were classified as being male are the ones who have the bigger brains. Those who are being classified as being female had the smaller brains. So it's not actually the qualitative differences in gray matter volumes, but it's rather just overall brain size, which is also very evident if we look at the characteristic of our training sample. This can be fixed in this particular case relatively easily by training on a sample that's not only matched for age, but also for brain size. And then looking at the model output, we see that the predictions are not anymore driven by overall brain size. So there is actually enough information in the brain, except brain size, to distinguish males and females, still with a high accuracy of roughly 0.87. So we have to be aware that models do reflect biases and limitations in the training data and in the labels, of course. And my final point, what can we do? These machine learning models often work basically as black boxes. So we get an output, we also get a good output in the sense that we can predict, for example, openness, personality score based on brain imaging data. In this case, brain imaging data from different movies and from different cognitive tasks. And we see that some tasks perform better at prediction openness than others. And to try to understand more what is driving these predictions, we can extract features that are particularly important for successful predictions. And we can see that these are different based on what feature set we are using. So those brain regions that predict openness particularly well are different when we predict from a language task or by showing a movie. And that is interesting. So we have some insight on what is actually driving these black box by looking at the most relevant features. But we really do still not know their effect in the individual. So which difference exactly in these brain regions makes individuals differently? And the question is really, are these explanations accessible for non-experts? So ultimately, the goal is to use machine learning-based predictions in a clinical setting, for example. And so far, it's just identifying most important features might not be enough yet to make these, to explain these black box well enough to actually explain them to patients and non-experts. So this is certainly a point that we still have to work on quite a lot. So we can interpret the model, but really interpret the results in a convincing way is much, much more difficult. So altogether, I think I've shown you some chances and some pitfalls. So we can, by using machine learning, find generalizing phenotype MRI relationships. We can apply these models trained on one data set to other data, so we can generalize, and we can do this for a variety of different phenotypes. And we can, by using clever feature space extraction, we can do a lot of different predictions from the same data set. However, we need to control for non-biological variability by looking at batch effects or controlling for batch effects. We need to be aware that the quality of the models depends on a lot of different factors, and that the models do reflect biases and limitations. And we really need to work a lot on and results interpretability to understand this black box better than we do currently. With this, I would like to thank you very much for your attention. Thank you very much, and bye.